So this is a webinar that we've been working on with Dr. Chris Fox from the Infectious Disease Research Institute for a little while. My name is Stephen Masidi. I'm the Director of Inside Sales and Applications for Microfluidics. And with me on the line is Dr. Chris Fox. And we've been working on a vaccine adjuvant formulation with IDRI for many years now. And Chris has generously offered to give this webinar for us and to provide a lot of information about his work in the area of vaccine adjuvants for global health. To start, I'm going to just give a quick overview of microfluidics and what we do here. And then it's going to be about 5 minutes, and then after that, Chris is going to talk about his presentation. We will have opportunities for questions at the end after Chris is done. A little bit about microfluidics. We were founded in 1982, and we've been producing high-shear microfluidizer processors. We have a very large installed base of machines globally. We were acquired by IDRI Corporation about 5 years ago, and within that time, we've been merged into a material processing technologies platform with 3 other companies. And our technology is used for many different applications globally, from chemical to cosmetics, but most of our business is in the biopharma markets, so that's what the focus of this webinar will be. And we have a vast experience of doing process development and formulation development with our customers. We have a very large applications database here, as well as we have a very experienced engineering team who are very familiar with all of the regulatory CGMP manufacturing protocols. So we have a lot of experience helping our customers pass those regulatory hurdles and bring their products to market. This is what we do best. Our primary applications are nano-emulsions and cell disruption, but we also do a lot of work with polymer nanoparticles, liposomes, do particle size reduction of different materials, as well as de-agglomeration. And for some applications, we can actually do molecular weight reduction. That's primarily for polysaccharides, but there are a couple of other applications where molecular weight reduction is the goal. We have a diverse product portfolio from the lab scale all the way up to the production scale. So we can offer many different processing options, including different batch sizes, and also different levels of cleanliness. We offer pharmaceutical-grade machines for processing both on small clinical batches all the way up to the production batches. And all of our machines are based on these fixed geometry interaction chambers. This is a schematic of a microfluidizer. They all use the same overview from the lab scale all the way up to the production scale, where there's a continuous process with an inlet reservoir, and then all of the material is pumped at constant pressure with these constant pressure intensifier pumps. And then they're pumped at very high pressures through these fixed geometry interaction chambers. Then downstream, there's a heat exchanger or other energy removal device to remove any temperature or energy that was imparted on the material during processing. These are custom pumps, and they're designed to work with complicated materials, either slurries with high solid loading or materials with high viscosity. They're very, very robust pumps. Then this is a schematic of our interaction chambers. We have 2 types of interaction chambers. The one on the left, this is the Y-type interaction chamber, and the one on the right, this is the Z-type interaction chamber. These chambers work in a similar fashion where you expose liquid streams to very high pressures and force them through microchannels. So the Y-type chamber has 2 microchannels. Material comes in through the top. It is forced to split into 2 streams, and then those 2 streams are forced to impinge upon each other. The high pressures cause very high velocities within these microchannels, and then those high velocities then cause very high levels of shear and impact. For the Y-type chamber, the impact is used for mixing. Since these 2 streams impinge, you get very high levels of mixing. Mixing is important for a few different applications. The primary one is nano-emulsion. When you generate nano-emulsions, you generate very small oil droplets with very high surface area. And you need to coat this surface area with a surfactant. In order to do that, you need to make sure that the surfactant is available at the surface, or else the emulsions will not be stable. You need very high levels of mixing in order to ensure that the surfactant is available. So Y-type chambers are typically used for emulsions and lysosome applications, things like that. The Z-type chamber, you get similar levels of shear and impact, but the impact is a little different. Instead of forming an impinging jet, the stream is forced to impact a wall. And during that impact, you cause particle breaking. So we use this for particle size reduction, deagglomeration, and cell disruption applications. And then the other nice thing about these microchannel designs is that they scale up linear. When we go to a scale-up machine, we can put multiple of these microchannels in parallel and achieve the exact same results on the production machine. Since you can process with the same geometry microchannel at the same pressures, you get the exact same velocities and therefore the exact same levels of shear and impact, and therefore you get the same particle sizes on the production scale that you were able to generate on the lab scale. So that's the benefit of our technology. We offer very high processing pressures, so we get very small particle size distributions. We have constant pressures and uniform geometry interaction chambers, so you get very tight particle size distributions, as well as very good batch-to-batch repeatability. And we also scale up very well. This is, our smallest volume machine. We can process volumes as small as 1 milliliter, and then this is our large production machine, which can process 80 liters per minute. And when you compare the two, you get virtually identical results. And then we have a very diverse product portfolio, so odds are we have a machine that is very suitable for your application. Depending on your cleaning requirement, as well as your flow rates and batch sizes, it is likely that we have a microfluidizer that's right for you. All right, so without any further ado, I'm going to hand this over to Dr. Chris Fox.
All right, thanks a lot, Steve. It's a great pleasure for me to present today, share a little bit about what we're doing in the field of vaccine adjuvants. So for my talk, I'll start off by introducing what it is we do here at IDRI, with a focus, of course, on vaccine adjuvants. And then I'll talk about 3 specific examples of formulations we've been working on with microfluidics technology, namely aqueous nanosuspensions, oil and water emulsions, and aluminum nanoparticles. And then I'll finish up by looking at some of our efforts to tech transfer the formulation and manufacture of these adjuvants to developing country manufacturers, and then where I think the field in general is headed. So IDRI has been around for over 20 years. We were founded by Steve Reed, who's currently the CEO. And, we started out by developing a diagnostic assay, a rapid test for leishmaniasis to replace the invasive procedure that Steve was familiar with from his 5 years in Brazil prior to founding IDRI. Since then, we've expanded to 125 employees, and we're largely funded by grants or contracts from the organizations you see listed here. And we're located in Seattle. We're a nonprofit, but we operate much like a biotech company in that we are focused on product development. So as I mentioned, we started out developing diagnostics, and we now have those on the market for neglected disease indications. Our vaccine and adjuvant program, we've developed both vaccine antigens as well as adjuvants that we've shared with partners, and we've done a total of 26 phase 1, phase 2 clinical trials with those components. And then we also have a drug discovery program for tuberculosis. And so those three activities are ongoing here at IDRI. It gives you an idea of the indications we're working on. So the pink shaded indications are where we have developed the antigens ourselves here at IDRI. And then the light blue indications are antigens from partners that are employing our adjuvants either clinically or preclinically. And we're also working on veterinary vaccine indications. So vaccine adjuvants, the simplest way to think about them as something that you add to vaccines to enhance or shape the immune response. And the kinds of vaccine adjuvants can be simplistically broken down into specific molecules. So I'll call those molecular mimics of pathogen molecular patterns, or PAMPs. And these are molecules that activate specific receptors on our immune cells, and then signaling cascades result in cytokine production. Some of these pathways are largely known. Others we're still finding out how they work exactly. Another class of adjuvants is really particulate formulations, and these are what I call geometric mimics of pathogens, either on particle size or other physical properties, which help the vaccine get taken up better or delivered more appropriately. In reality, most adjuvants don't belong solely in either category, but they're combinations, where we'll often take a immunostimulatory molecule, like you see on the left there, the TLR4 ligand. We'll formulate that in some kind of nanoparticle, and then that's the finished adjuvant. So why adjuvants? There's a lot of benefits we think that adjuvants are bringing to the vaccine field and will bring in future. Let me just highlight a few of these. First of all, for indications where the current global vaccine supply is unable to cover the entire indicated population, so for instance, pandemic flu outbreak, where the world really couldn't manufacture enough vaccine rapidly to cover the entire population, adjuvants can facilitate dose sparing, meaning a reduced amount of vaccine can give an equivalent response with adjuvants as the higher amount of vaccine. So this becomes very important in those situations. A similar benefit would be a reduced number of immunizations that could be facilitated by inclusion of adjuvants. Regarding indications where there's a challenging immune response that needs to be elicited, and it's not just antibody-based like most existing vaccines. So we're talking T-cell-based indications like TB or malaria or perhaps HIV. These require potent adjuvants to generate this type of cellular immune response. And so you'll see more and more, I think, action here in the field from adjuvants. Also, therapeutic vaccines in a similar vein that require a more complex and potent immune response. Adjuvants are really finding applications here. Now, there's certain populations that, either because of immune senescence in the elderly or in very young children where the immune response is not completely developed, they don't respond so well to some vaccines. Adjuvants can change that paradigm and promote better responses in these populations. Also for pathogens that undergo drifts or shifts, such as influenza, adjuvants can broaden our immune responses to protect against heterologous strains, even that aren't present in the actual vaccine. And so this is a huge benefit as well. And finally, changing the timing of our immune response, mounting a faster immune response or one that is of longer duration, are also things that adjuvants can do. So this is why we're interested in them. We think they're a real game changer in the vaccine field. And the one thing that we could really stand to learn more about is how each adjuvant is working biologically. And there's been tremendous progress in the field over the last decade or so, but there's still much to be done. In this diagram, I've highlighted those two classes of adjuvants I mentioned earlier. So the immunostimulatory molecules such as TLR ligands, we bind to known receptors called TLRs, which then activate a signaling cascade, resulting in cytokine production, which can then drive the resulting immune response. As for the particulate formulations, they may promote vaccine antigen uptake, so you'll get increased uptake or more efficient processing in the antigen-presenting cell. Particulate formulations may also activate a complex called the inflammasome, which itself results in cytokine production, which in turn can recruit immune cells to the site or to the lymph node. And together, these responses then eventually result in either cellular and or antibody responses of specific qualities such as Th1 or Th2. And the type of response you want depends on the disease that you're working to vaccinate against. So this is a simplified history of adjuvant development. And I'm borrowing this concept from Derek O'Hagan, an expert in the field, where we have this timeline showing the first adjuvant to be employed in human vaccines, which was really aluminum salt. We call it alum for short. And for many, many years, this was really the only adjuvant in licensed vaccines. More recently, we've been seeing different classes of adjuvants approved in Europe and the U.S. now. And that includes oil and water emulsions in flu vaccines. It includes TLR4 ligands in addition to alum for certain viral vaccines. And once again, we're seeing these same vaccines approved in the U.S. as well now. So it's an exciting time for adjuvants. I think in the near horizon, there's even more promising vaccines that are being developed. For instance, GSK's shingles vaccine completed phase 3 trials recently with an almost unbelievable high protection rate, even in the very elderly. And so that's a liposomal adjuvant containing 2 different immunostimulatory compounds. I mentioned the oil and water emulsion adjuvant. Canada last year approved a pediatric seasonal flu vaccine for very young children based on that principle of adjuvants increasing immune responses in young kids. So, for instance, a seasonal flu vaccine without adjuvant, you might expect something like 40% protection in this age group, meaning 6 months to 72 months of age. But with the adjuvant, that climbs up to over 85% vaccine efficacy. There's a malaria vaccine that's being rolled out in pilot stages with this same liposomal adjuvant I mentioned earlier in the shingles vaccine. And there's another immunostimulatory compound, a TLR9 ligand, which has also completed phase 3 testing. And so we'll be watching this closely to see where that goes. Overall, I think, very positive trajectory for the field. So specifically here at IDRI, we work on formulating various adjuvant compounds. So some of the molecules we work with are listed on the right here, various TLR ligands or saponins or combinations thereof. And we've developed some basic formulation platforms that we adapt according to the physical chemical properties of each TLR ligand. And so I'll be discussing some of these today. We also offer services to the field in general. So we're interested in partnering and licensing technologies based on adjuvants and vaccines. We also have a cGMP manufacturing suite suitable for manufacturing and still finishing material for phase 1-2 clinical trials, whether it's vaccine-based or some other drug-based application. So I'd be happy to entertain any interested parties about using some of these services. Okay, let's get into a little bit of data. So the first example I'll talk about is an aqueous suspension that we developed here. We call it GLA-AF. GLA, the structure you see there, it's a synthetic molecule. It's a TLR4 ligand, which is a mimic of natural lipopolysaccharide. And the idea here is the pathogen-associated molecular pattern. It induces a potent signaling cascade through TLR4, but it's insoluble due to its hydrophobic structure. So you can't just drop this into a vacuum and run with it. It has to be formulated so it can go into solution. So we have various formulations for this molecule, one of which is this aqueous nanosuspension. It's really a simple formulation that consists of mixing the GLA in organic solvent with a helper lipid, such as a phospholipid, like BPPC, which is what we use here, and then evaporating that off and hydrating and providing some energy input. Traditionally, we had used water bath sonication for that energy input, and that resulted in nanoparticle sizes, which we then mix this type of formulation with a vaccine antigen, and that's the complete vaccine. So this has been done for other compounds as well, namely a naturally derived TLR4 in the HPV vaccine on the market. We've also most recently done this with a TLR7/8 ligand called 3-MO52. You can read about that here. I'm not going to talk about that today because I want to highlight with GLA-AF the manufacturing process development that we performed. And I'll just mention this formulation has been in clinical testing with various vaccines, namely pandemic flu and hookworm, via different routes. It's got a good safety profile. But we were manufacturing this by sonication, and the larger the scale we went to, the longer this would take. So at the 1 liter scale, we were sonicating for approximately 4 hours at a high temperature, and this just became undesirable and unworkable really as a manufacturing process. And that's when we looked towards microfluidization to see what improvements could be made. And so on the right here, you see the different pressure that we microfluidize the formulation at, the number of passes here on the other axes that it would pass through the interaction chambers that Steve described, and then the resulting particle size of the nanosuspension. So as you might expect, with higher pressure and higher numbers of passes, you get smaller particle size. And really after about 5 passes, the reduction in size isn't much. So this became a significant improvement in our manufacturing process. We were getting equivalent or smaller particle sizes than we got with sonication, and yet it was an order of magnitude less time to get to that point. We saw excellent recovery of the GLA, the active ingredient, and so we selected the 30,000 PSI conditions to move forward, and we've gone up to the 1 liter scale on our microfluidizer and seen a good result. Now, of course, it's not just the physical chemical properties, but the question is, do we retain the bioactivity with the different manufacturing process? So our first look at that was an in vitro assay. So we take either a macrophage cell line on the left here or human whole blood. We stimulate the cells with the adjuvant formulation. So we made 2 batches each of a sonicated formulation or a microfluidized formulation. And then we measured the production of a cytokine MIP1 beta from the cells after they've been stimulated with the adjuvant. This is a cytokine that's responsible for attracting various immune cells. And what we found was overall a similar profile regardless of the manufacturing process of the formulation. if anything, we saw a little bit tighter reproducibility with the 2 microfluidized batches compared to the 2 sonicated batches. But certainly no loss in bioactivity on this in vitro assay. And then we did an in vivo study. We immunized mice with a recombinant malaria antigen, PBCS2, and combined with the vaccine adjuvant or alone. And what we saw was, regardless of whether the adjuvant was sonicated or microfluidized, a similar antibody response after 2 immunizations. These were statistically higher than antigen alone, as you'd expect. And then as far as T-cell response, antigen-stimulated splenocytes produced interferon gamma in the adjuvanted group, but not so much in the antigen alone group. So overall, we're happy with this because we had a more efficient manufacturing process for this adjuvant formulation without detracting from the bioactivity that we had seen previously. So in my next example, I want to discuss an oil and water emulsion, and this is known as FE, stands for stable emulsion. This is really the flagship adjuvant formulation platform for IDRI. We've been making these for many years now. And they're based on squalene, so that's a biocompatible oil. They're processed by microfluidization to generate approximately 100 nanometer diameter droplets. These are monodispersed, as you see in the dynamic light scattering data on the left here. And these are quite stable, refrigerated. We get several years stability. And we've made many GMP batches now and provided those for clinical testing. The emulsion itself is an effective adjuvant for certain vaccines, such as killed inactivated influenza vaccines. For other highly purified recombinant vaccines, we'll often formulate the emulsion to include another immunostimulatory compound, such as a TLR ligand. But my comments today are focused on just the emulsion without an additional TLR ligand. So these are highly interesting adjuvants. Number one, because next to alum, which has been in well over probably 1 billion human doses, emulsions are the most widely used adjuvants in human vaccines. So there's something like 200 million human doses that have been administered with oil and water emulsions. As I mentioned earlier, they can really enhance responses in the young population or in the elderly. And so that really ups the influenza vaccine efficacy in those populations. Now, since 2014, we've been working with BARDA from the U.S. government to increase the manufacturing capacity of our emulsion adjuvant, FE, here at IDRI. So rather than making thousands of doses like we've done previously for phase 1 testing, the idea was to up our capacity to be able to make millions of doses in the event of a pandemic. So we knew when we were given this charge that we were going to need to make some efficiency improvements in our manufacturing process in order to generate this amount of adjuvant in our small GMP facility. So this was the previous process. It involved several steps. Number one, we had 2 separate phases to prepare, an oil phase and an aqueous phase. The oil phase involved a sonication step. There was then a high-speed overhead batch mixing process followed by microfluidization for about 16 passes, and then a sterile filtration step. So looking at each step in this process, we tried to identify efficiencies. And one of the things we did was remove the separate phase of preparation and just put it all into one, what we call the one pot method, and remove the sonication step. Replace the overhead batch mixer with an inline mixer, which we can then connect directly to the microfluidizer. And then we upped the concentration of oil so that we were making a more concentrated formulation, which could then be diluted following manufacture to the appropriate concentration. And these efficiencies improved about sevenfold what we were able to do. But in consultation with Microfluidics, they indicated that actually upping the concentration of oil might result in the need to do fewer passes on the microfluidizer. This was counterintuitive to us, but sure enough, instead of having to do 16 passes, we find we can get away with 5 or 4 passes now with this new method. So here's what it looks like schematically. So instead of 2 phases, there's the 1 pot approach, inline mixer, the 5 passes now, and then the filtration and later dilution prior to use. So one of the things that made this process improvement possible was the scalability of the microfluidics equipment. So we have all 4 of these microfluidizers here at IDRI, and I'm showing the typical batch sizes that we make with each model. You can see the increased flow rates as the models get larger. Now our large capacity will obviously be done with this large 7250 microfluidizer. But the process improvements I talked about were done on smaller models. And the reason we felt confident in doing that was this data on this slide. So here you're seeing particle size from the nano-emulsion process on each of the 4 different microfluidizers we have here at IDRI. And the 5 passes I mentioned, you can see the particle size go down. Very similar performance in each microfluidizer. And so we were very happy to see this. And, I think as Steve indicated, it's due to the approach of using these parallel interaction chambers, I think, which allows this translation to the larger scale. But we didn't want to base all our decisions on the small scale. We certainly wanted to do some pilot batches. So these are some images of when we went back to Microfluidics facility near Boston, their pilot facility. This is Steve helping us make a 20-liter batch of FE. And as I mentioned, the model performed quite nicely, and we're now working on installing the equipment here at IDRI and implementing the more efficient method. So the last case study I want to transition to now is this is a newer project for us. This is earlier stage and certainly not in GMP manufacture or clinical testing. But this is based on the oldest adjuvant, which is alum. But we tried to take another look at alum and improve, if possible, on its properties to make a new formulation, which we're calling Nano-alum. So, as I mentioned, alum's been around for a very long time. We know a lot about how it works. There's still work to be done here. But it's generally very well accepted by regulatory authorities, and it's known to enhance vaccine responses. Some of the challenges with alum is it's a heterogeneous mixture of particle sizes. So you can see in the cryo-EM images down here, these sort of loose aggregates of these rod-like, semi-crystalline particles. And really you end up with a large range, anywhere from 100 nanometers to 10 microns of these aggregates. And anyone who's worked with alum that's listening will know that you can see sedimentation quite rapidly in a glass vial if it contains alum. And that's because of the large particle size of the material. So this makes it impossible to filter through 0.2 micron membranes. Therefore, it has to be autoclaved to be sterile. We also felt like if we could get more control over the particle size, that it might offer us some bioactivity advantages if we could have a formulation with a tunable particle size. So what we did was we started with a commercial alum formulation, a microparticle alum called hydrogel. This is widely used in the field. And we knew from previous experience that if you sonicate or microfluidize with hydrogel, you will reduce the particle size, but it quite quickly re-aggregates into the original microparticle size. So our idea was to mix this alum with what we would call a sizing agent or a stabilizing agent. So what we ended up with was either pegylated phospholipid or polyacrylic acid. And so these polymer-based compounds, if they were present with the alum when it was microfluidized, they would allow us to maintain that nanoparticle size reduction. And so I'm showing you, this is another image of microparticle alum before it's processed. And then when it's processed in the presence of the pegylated phospholipid, you see these, we keep these sort of rod-like structures, but they don't aggregate anymore. They're individual particles. And we can tune the size based on the number of passes, as you see here on the right in the microfluidizer, or the pressure, or if one is interested in getting larger particles, say half micron, you can do a lower energy method like high-speed mix. In any case, we end up with particle sizes that now are small enough that they don't sediment. So the data I'm showing you on this slide is a technique called laser scattering optical profiling. And basically, you're shining a vertical laser light through a cuvette and monitoring the percent transmission through whatever formulation's in the cuvette. And so the time course I'm showing here, the red scans are the initial scans, and then moving to green are the most recent scans. This was over 4 hours with scans taken every minute. And what we saw with unprocessed alum was that you get very rapid sedimentation. So in other words, the top of the solution clarifies, you get more light through, while the bottom of the cuvette, due to sedimentation, less light comes through. On the other hand, the nanoparticle alum, you essentially see a constant transmission profile over time throughout the cuvette. And this particle size is maintained long term. So these formulations on the left here is the polyacrylic acid stabilized nano-alum. On the right is the pegylated size alum. And we see at least 6 months. We're still monitoring this. We think it's going to go longer term. For the pegylated version, we're seeing 12 months now of physical stability. We've used atomic emission spectroscopy to show that we're not losing any aluminum, so we're recovering all of that after processing. And the big one is we can now filter through 0.2 micron membrane that removes the need for autoclaving the formulation. Okay, but how about bioactivity? So this was interesting for us. We found that the adjuvant activity of nano-alum was highly dependent on the excipient modifier that we employ to maintain particle size. So our first studies were done with a 5,000 molecular weight pegylated lipid of various sizes. And almost in every case, we found reduced adjuvant responses compared to unprocessed alum. So we went back to the drawing board, used some different molecular weight polymers, and we ended up with 2,000 molecular weight PEG with DFP as the lipid, and an additional TLR4 ligand similar to the GLA I described earlier. This one's called FLA, and then a recombinant TB antigen that we developed here at IDRI called ID93. And we found that mice immunized with this formulation were able to maintain the appropriate immune response compared to unprocessed alum. So what I'm showing here is what we call a Th1 type immune response. These are CD4 T cells stimulated with antigens, and they then produce the cytokines shown. And what we're looking for is upregulation of interferon gamma, TNF, and CD154. And you can see in orange the unprocessed alum with the TLR4 ligand, and then in red is the Nano-alum now. So if you compare the red to orange in the indicated cytokines, we're seeing pretty good comparability. IL-5 is actually something we don't want to see. It's a Th2 indicator. In fact, we see less in the Nano-alum than in the M-process alum. So the Nano-alum is at least as good, potentially a little bit better in this case, as far as bioactivity is concerned. So in other words, we've improved some of the manufacturing characteristics of the alum without losing bioactivity. Now, for the polyacrylic acid Nano-alum, we've got some data coming in on this formulation now. And this is without a TLR4 ligand, so it's just the alum and the vaccine antigen. And we're finding that we're getting upregulation, surprisingly, of these Th1 type cytokines, even without a TLR4 ligand, in the Nano-alum formulation in gray. Whereas the formulation that only contains unprocessed alum or just the PAA alone, we're not seeing appreciable responses. So this is very encouraging, and appears we're enhancing now adjuvant activity with this particular Nano-alum. So to finish up, I wanted to put this research and development in context of our mission to really provide increased access to adjuvants throughout the world and to transfer adjuvant technology, manufacturing, formulation technology to places where it's really needed the most. And so this is dealing with more than just the sort of scientific aspects that I've covered so far, but really the whole product development perspective, whether it's raw material supply, IT, regulatory considerations, and really fill a gap for the public sector and provide them this expertise in order to develop products. So on the map here, I'm showing some of the institutes we've worked with around the world. I just want to highlight a few examples. So I'll start off with Genova Biopharmaceuticals in Pune, India. You can see they've got a 110P model there. They also have a biopharma 110EH model. And we started working with them in 2010 and transferred the manufacturing process for the emulsion formulation with the TLR4 ligand GLA. And we've also transferred other formulations since then to them. And they've made very exciting progress with help from the Malaria Vaccine Institute and the Department of Biotechnology in India. They actually constructed a new building, a vaccine formulation center, to do this work. And I just received an email from my colleague there, Swarnendu Kaviraj, yesterday, saying that they finished GMP manufacture of the GLA-AF adjuvant earlier this year. And the phase 1 clinical trial with a malaria antigen, also manufactured in India, has initiated. They have 14 subjects immunized so far. So this is a great success story of a vaccine made in India for India, and it's a complex vaccine. It contains adjuvant components and antigen components. Another example is in Romania with the Santacadino Institute in Bucharest, where we transferred the emulsion manufacturing process to them for their pandemic flu vaccine program, and they afterwards demonstrated they could independently make the adjuvant, and it was effective in ferret challenge studies. More recently, we've been working with Instituto Butantan in Sao Paulo, and they are also working on pandemic flu, and so we've been working with them on adjuvant technology. And then in Cape Town, South Africa, with Afrigen Biologics, where they are now making research batches also of the emulsion formulation of GLA for future TB vaccine testing in South Africa. Now, besides actual manufacturing tech transfer, we've been working on higher level training of doctors and researchers on the benefits of adjuvants and their appropriate use. And so we've taught a course at CMC in Vellore, India, for 5 years. And we've also taught an online course sponsored by the Carlos Slim Health Institute. It's broadcast in Central America. And then we try to get the word out, of course, through publication. Here's the Romania example. It's published in Vaccine. Also, we just recently published a book really focused on practical aspects of adjuvant manufacture. And all this with the idea that to really increase know-how and access for researchers and product developers around the world. So in the future, I think we can continue to see this acceleration of adjuvant technology in licensed products. Some of the things that we're watching and are participating in include developing thermostable adjuvants. So this is the emulsion lyophilized into a cake to be stored at room temperature or elevated temperatures for multiple months. Also, adjuvants that can facilitate alternative routes of delivery. This is data from an intranasal administration of a combination adjuvant from our lab with a diarrheal disease antigen called amibiasis from the University of Virginia. We're seeing protective efficacy. This is a very complex immune response that's required to generate this. And then also in immunotherapy applications, we're finding that adjuvanted vaccine combined with chemotherapy approaches can reduce time to cure. This is a leishmaniasis patient suffering from cutaneous leash. And we saw reduced time to cure in these patients, excuse me, if they were given vaccine along with chemotherapy. So with that, I'll conclude and leave some time for questions, Steve. I'll leave the conclusion slide up and turn the time back over to you.
Great. Thank you very much, Chris, for that excellent presentation. We are very happy that we've been able to work with you for the past few years and are very excited about all the work that you do globally. And with that, I will open it up to questions. So if anybody has any questions, feel free to type them into the chat window. I see we already have one. Can you buy the adjuvant book online?
You can. It's available online now.
Okay, great. And do you have a bibliography for your applications using the microfluidizer technology?
That's a good question. This presentation is probably as close as we've come to that. We try to publish everything we do as a nonprofit institute, and so more info can be found there. But I think that's about the extent of a bibliography compilation.
Okay, great. Are there any other questions? One question that came prior to the webinar was around the pre-formulation, and Chris did a pretty good job addressing that when he talked about his one-pod technique, but I'll touch on that a little bit more. With the microfluidizer processor, the mixing levels inside the interaction chambers are so high that typically, as long as you are able to get all of your ingredients dissolved and into your formulation in some sort of a somewhat homogeneous method, combining them inside the interaction chamber is actually pretty typical, and we have noticed that formulation work gets a lot easier when you're using our technology. Another question we had was around the types of interaction chambers that you use for these applications. Chris, I know that you're using a Y-type interaction chamber for the emulsion. Do you know what type of interaction chamber you're using for your dispersion?
Yeah, in all these examples, we're using the Y chamber followed by a Z chamber for back pressure.
Okay, great. And then another question is, have you ever considered mixing the alum adjuvant formulation with the nano-emulsion formulation?
Yeah, that's a very interesting concept. We have done that. It forms what's called a Pickering emulsion. In other words, an oil that's stabilized by a solid phase stabilizer rather than a typical emulsifier. And so it can be done. It's interesting physically, but we haven't seen a lot of bioactivity benefits from that yet. I think more work needs to be done, but so far that's our consensus.
Okay, great. All right, if there are not any other questions, I'd just like to take this opportunity once again to thank Chris and conclude the webinar, unless Chris, you have any further comments.
No, just thanks a lot, Steve, for the opportunity, and look forward to hopefully working with some of the listeners in future.
Great, yeah, and once again, our contact information is on the screen. Don't hesitate to reach out to us if you have any further questions. Thanks again.